Senior Global Marketing Manager here at NanoString, your NanoNews correspondent, coming to you live from booth 1719 at AACR 2022 in the Big Easy in New Orleans, Louisiana. So let me walk you through the booth and give you a little tour. Come on. So we're most excited about our newest product launch in the nCounter analysis system product line. The nCounter Pro utilizes the same simple digital barcoding technology that allows you to profile 800 plus genes overnight with only 15 minutes of hands-on time and gets you publication quality results in less than 24 hours. But it has an updated operating system, hard drive and data encryption to keep your data secure, enable a data security ecosystem, and help support a 21 CFR Part 11 environment, which is very important for auditing and tracking purposes in the biopharmaceutical industry. In addition to that, we have an updated Hallmarks of Cancer publication that recently came out, and you can download that publication by scanning the QR code here. And this will also take you to the NanoString website where you can learn more about our nCounter Pan-Cancer Panel Collection, which is a series of 770 plex gene expression panels for human and mouse that incorporate many of the pathways that are involved in the hallmarks of cancer. And if you come over here and follow me, I'll introduce you to our spatial biology platform, GeoMx Digital Spatial Profiler. So the GeoMx Digital Spatial Profiler allows you to spatially profile in multicellular structures and tissue architecture in FFP or fresh frozen tissue sections up to the whole transcriptome and hundreds of proteins. We can even simultaneously profile RNA and protein from a single tissue section with a workflow called spatial proteogenomics. The GeoMx instrument has content for immunology, neuroscience, and oncology, and we also have the ability to customize probes for your own protein or RNA markers. And to learn a little bit more about the power of the GeoMx Digital Spatial Profiler, I'll take you over here to our interactive, and I will introduce you to the Spatial Organ Atlas. So the Spatial Organ Atlas is an initiative at NanoString that allows us to map the architecture of organs on the molecular scale. We have profiled a number of organs, both in human and mouse, and we have used the GeoMx Digital Spatial Profiler to profile the whole transcriptome in different tissue structures and segments and different organ structures within each organ. For human, we have the brain, lymph node, pancreas, colon, and kidney. And for mouse, we have the brain. And later this year, we're going to be releasing a mouse embryo for fetal development. This is on our website. You can go to our product tab under GeoMx to the Spatial Organ Atlas. And actually, there's downloadable data sets. You can click on each organ, and there's downloadable data sets. In addition, there is a Minerva story, which guides you through the data and allows you to visualize different parts of the tissue. And it actually is a story that walks you through the content and the different tissue structures that are in the brain and how we've segmented the brain based on different substructures as well as cell types. And if you come over here and follow me, I will tell you a little bit more about our other spatial biology platform, the CosMx Spatial Molecular Imager. So the CosMx Spatial Molecular Imager is actually going to be launched later this year, but it's already available through the Technology Access Program where you can send us samples at NanoString. So the CosMx Spatial Molecular Imager allows you to profile RNA and proteins in single cells in FFP tissue sections. So not only can you look at single cells, but you can actually look inside cells at subcellular structures and organelles within individual cells. We have the ability to profile 1,000 RNAs as well as 100 proteins. And combined with the power of geomics, as well as the nCounter for bulk profiling, we have a whole ecosystem for biomarker discovery and gene signature development. We actually have a poster that's being presented at AACR on the fact that we've been able to profile 100 proteins in FFP tissue sections at the single cell level with the Cosmic Spatial Molecular Imager. Come by the booth at the exhibit hall. We are booth 1719. In addition, we have a Spotlight Theater presentation that's happening on Tuesday morning in Theater A at 10 a.m. Joe Beecham, our Chief Scientific Officer and Senior VP of R&D, will be presenting about both the geomics and the cosmics spatial biology platforms that allow you to create a true spatial biology ecosystem in your lab. In addition, we will have Arutha Kulasinghe from Queensland University of Technology talking about a study that they did with COVID-19, spatially profiling tissue sections. And then we have Hargeeta Kaplan from Altreca Inc. And she will be talking about looking at T-cell receptor diversity within tissue sections spatially. So thank you so much for joining me on a brief tour of the NanoString booth. We hope to see you in person here at AACR 2022. Hello everyone, it's your favorite NanoNews AACR 2022 correspondent with NanoString, Elizabeth Schneider, Senior Global Marketing Manager, and I'm here today with nCounter Product Manager Brianna Murphy, and we're excited to be launching a new nCounter analysis system, the nCounter Pro. So Brianna, for those of the people watching who aren't familiar with the nCounter, could you tell us a little bit about the nCounter technology and what makes it different from RNA sequencing or qPCR?
Yeah, absolutely. So this is the nCounter Pro, which is a mid-plex gene expression profiler that uses direct detection in only 15 minutes of hands-on time to deliver gene expression profiling data for many different kinds of samples, including difficult sample types such as FFPE. And sometimes people are confused about how it's different from sequencing, but it actually uses unique chemistry using direct detection of the molecules that are present in the samples. So there's increased reliability and lower variability in your data, since you don't have to have any amplification, reverse transcription, or cDNA conversion, which really produces data that you can feel confident that you're going to be able to publish with very easily.
Excellent. And then what about the analysis, like compared to, say, the bioinformatic pipelines that are required with NGS and sequencing? How is the nCounter data analyzed?
That's actually one of my favorite things about the nCounter platform, is that you're able to have access to all these intuitive analysis platforms. Really, there's a lot of different options based on what your needs are. But my favorite is using the Roslyn platform, which we have a collaboration with the team on, which you're able to do cloud-based analysis using a suite of intuitive analysis tools where you can visualize the data on a web page. You don't have to add any programming knowledge beforehand to be able to use it. And you can export really beautiful figures to help your publications and being able to draw insights from your data very quickly.
And as far as I understand as well, you can also collaborate in a collaborative space with colleagues who are across the globe.
Which is relevant today in a hybrid work environment, right?
Yeah.
So you don't have to be co-located to be able to look at data. And you're going to be able to log in and access that data and then share it with anyone who you want to give access, or keep it from people who don't want to have access as well.
That's great. So can you tell us a little bit about what makes the nCounter Pro unique? What are the features of this new nCounter system?
Right, so we're really focusing on how simple, secure, and reliable this new system is. So it's simple because you can analyze your results in only 24 hours. It has a very simple workflow on bench, which makes training new operators very easy, which is relevant in today's labor market. It is reliable, so your data is going to be publishable. There we have over 5,300 peer-reviewed papers already on the market that show how trusted this technology is. All the things that we talked about the nCounter workflow really help produce reliable, robust data that publishers and papers really trust. And it's secure because it enables a 21 CFR Part 11 environment. So you know that you're going to be able to work with a system that's going to protect the integrity of your data and fit within your institutional IT requirements as well.
Yeah, and I seem to recall that there's an updated operating system, hard drive, and data encryption that comes with this instrument.
Yeah, so this is going to be a completely locked down system. So any IT professional who's looking at this when you're going to purchase a system is going to feel confident that it's going to be able to support an overall data security ecosystem.
Excellent. So can you tell us what these two different boxes are? I see there's two systems sitting here next to each other.
Right, so when you go to set up your reaction, which is a simple 5-minute pipetting step, you'll hybridize your samples overnight, and then the next day you'll take your samples onto the prep station here, which is a liquid handling robot that will purify your samples, remove any of the extra code sets, and prepare it for scanning on the digital analyzer here, which will count the number of hybridization reactions with our unique molecular coded barcodes, and then be able to analyze that using whatever data analysis system that you have.
Excellent. And can you tell me a little bit about content? So if someone wants to study human or mouse or rat, oncology, immunology, neuroscience, do we have any off-the-shelf reagents for someone who wants to do an nCounter assay?
Yeah, absolutely. We have pre-made panels that are ready to ship in all disease areas. One of our newest ones is going to be the cardiovascular panel coming soon. So being able to look at different physiology pathways. And if we don't have a panel that's available, we have the option to spike in up to 55 custom targets into one of our existing panels, and we have other options available as well if someone wants to have a custom solution that we don't have an off-the-shelf option for currently.
Oh, excellent. And I'd also like to point out that I see that there is an updated Hallmarks of Cancer publication that came out through AACR, and some of our first panels that were released was our pan-cancer panel collection, pan-cancer pathways, pan-cancer immune profiling, and pan-cancer progression that are panels for human and mouse. In addition, we have our pan-cancer IO 360, breast cancer 360, and tumor signaling 360. And all of the pathways included in the Hallmarks of Cancer are included in this comprehensive portfolio of panels for cancer research. And for any of the additional Hallmarks of Cancers that have been updated, if any of those pathways are not in our panels, you can spike it in using that Panel Plus feature of up to 55 genes that Brianna mentioned. If you come by our booth, it's 1719 here at AACR in the exhibit hall. You can scan here this QR code to download a copy of the updated Hallmarks of Cancer publication. So thank you so much, Brianna. It was a pleasure chatting with you.
Thank you, Elizabeth.
Yeah, and we hope to see you soon at our booth. Hello, everyone. I am your NanoNews correspondent coming to you live from the NanoString booth at AACR 2022 in the Big Easy in New Orleans. My name is Elizabeth Schneider, Senior Global Marketing Manager here at NanoString. And I have the pleasure of interviewing today Will Wong. He is a physician scientist at Massachusetts General Hospital. So Will, it's a pleasure to have you here. Could you start out by telling us a little bit about your work?
Absolutely. So I do interdisciplinary research between Mass General, the Broad Institute, and MIT on GI cancers, with a particular focus on pancreatic cancer. And what our team is very interested in is understanding therapeutic resistance at high resolution using single cell and spatial tools.
Excellent. That's very interesting. So I hear that this year you were selected as one of the AACR Next Generation Stars. Can you tell us a little bit more about that?
That was a really nice surprise to find out that I would have the opportunity to speak at a major symposium at AACR. And what the Next Gen Stars program does is provide a platform for young and up-and-coming investigators to speak to a broader audience about their research.
Excellent. And I hear that later today in the morning you're going to be presenting on your work?
Yes. So at 10 a.m. I'll be presenting our work at the Nerves, Cancer, and Immunity Symposium.
Excellent. Well, I hope everyone who is here and watching this is able to attend, or everyone who's virtually here can live stream the event. So everyone is talking about spatial biology. It's the hot buzzword at the moment. So what excites you the most about the field of spatial biology?
I think there's many things about spatial biology that are generating this well-deserved excitement, and I think what we've realized is that studying cancer intrinsic or cancer extrinsic signals on their own is really not enough to really understand why cancers evade therapies and why they progress and disseminate. And pancreatic cancer is one of those examples where the outcomes are far from where we need them to be. And that dynamic interplay between cancer cells and the various components of the tumor microenvironment can really only be studied with high fidelity using spatial tools, because you need to know which cells are not only near each other but likely interacting in a way that promotes tumor progression.
Excellent. And so I understand that you're currently using the GeoMx Digital Spatial Profiler. Can you tell us a little bit more about how you're using it?
Yeah, so the GeoMx Digital Spatial Profiler was very ideal for our studies on pancreatic cancer, because we are using clinical samples, and some of the historical samples are FFPE based, and the fact that the GeoMx platform was built on FFPE was very helpful. The second reason it's very helpful is because it has whole transcriptome, up to whole transcriptome coverage, as well as high-plex protein coverage. And what initially we had discovered a novel resistance mechanism or program actually using dissociated single cell data. And we wanted to put that in context, as I alluded to before, of multicellular communities and how various cells were interacting to potentially mediate that therapeutic resistance we observed. So we needed whole transcriptome coverage because a lot of the genes that define this novel program are not curated oncology genes that you'd find in a panel. And secondly, we had clinical FFPE specimens, so between those two things we were able to really seamlessly integrate GeoMx and our single cell, single nucleus RNA-seq into a compatible workflow, and then really map some of those insights we found in the dissociated setting onto the tumor architecture.
Oh, that's really exciting. So what do you think the future holds for spatial biology?
I think it has immense potential. I think, I obviously focus on oncology, so I can speak most to it, but I think in many areas of pathology, across the spectrum of human and animal health, I think we're moving into a phase where we can do basic discovery, like I alluded to, but we're also starting to see hints of how we can translate these findings into the clinic as biomarkers, as molecular stratification tools with clinical trials, as an aid to augment pathology, which is already plays a critical role in everything we do in the hospital setting. So I think all of those things will be very exciting, and I think, also taking a step back, just the fundamental biology that we're learning. One of the things I'm going to speak about at my talk in a couple hours is the dynamic between the peripheral nervous system and cancer cells, which has really been pretty understudied, but it's quite intricate and pervasive in many peripheral cancers like pancreatic cancer. And being able to study that spatially, there's really no other way, right? Because nerves are extremely asymmetric and elongated cells, and their interactions are myriad with other cells, and really you need that spatial context to be able to study it. So I think we're going to also unpack and uncover a lot of new basic biological insights with spatial technology, as well as the translational piece I mentioned first.
Excellent. And also, as you know, we do have the ability to spike in custom markers for RNA into a geomics assay, into the whole transcriptome. So you mentioned cancers of the gut. I'm hearing more and more in the literature about how the microbiome plays a role. Are you thinking about perhaps spiking in any targets for, different flora that is part of the microbiome and using that in the context of your larger studies?
I think that would be very neat, and it's definitely something that is not my team's direct expertise, but some of our collaborators and some of the questions we're interested in do relate to that, how the microbiome and the immune system and the nervous system interact. And so I think for sure in the future that would be something that we would consider.
Excellent. So as a final parting question, I know that everyone has been sad not being able to attend conferences in person for the last 2 years in the pandemic. And this year at AACR 2022 is one of the first opportunities that we're able to get in person together. So what are you most excited about as you're attending this conference?
It's true. I mean, it's been a long 2 years, and I think virtual conferences, as great as they are, I think the part they don't replicate is clearly the networking, seeing, old and new colleagues and this interaction that we're having and learning about all the kind of amazing technological developments that are happening in industry that really help us do our academic work in a very synergistic fashion. I think all those things we can't really replicate in a virtual format. So I think I'm very excited to have these face-to-face conversations, to establish new collaborations and renew old ones as well.
Excellent, and I would definitely agree with you. It's great to finally be in person. I was getting cooped up working from home in my apartment. So thank you so much, Will. It's been a pleasure chatting with you. And thanks everyone for joining us and watching on social media. And if you're here at the meeting, please stop by the NanoString booth. It's booth 1719 in the exhibition hall. And we will be having a Spotlight Theater presentation in Theater A in the morning at 10 a.m. on Tuesday. Thanks everyone. Thanks, Will.
Absolutely. Thank you.
Hello everyone. Elizabeth Schneider here, Senior Global Marketing Manager at NanoString. We're coming to you live from AACR 2022 in beautiful New Orleans, Louisiana. I have here with me today a data scientist from NanoString. This is Tyler Hether. He's presenting right now his poster that we did in a collaboration with the Memorial Sloan Kettering Cancer Center, the Parker Institute for Cancer Immunotherapy, University of Pennsylvania, and Dana-Farber Cancer Institute. And Tyler is here to tell us about this poster, which was a multi-institution examination of concordance in spatial transcriptomics using the GeoMx Cancer Transcriptome Atlas. Wow, that was a mouthful. I'm so glad you're here, Tyler, to walk us through this. So what was sort of the impetus for doing this study?
Well, so with GeoMx, you're able to profile like discrete physical locations on a tissue, and you're able to interrogate the gene expression profile across multiple regions of interest, or ROIs. And we could do this across lots of studies, and it's been very helpful to elucidate markers and drivers of cancer. And with GeoMx, we have now over 100 publications since its debut in top-tier journals. One natural follow-up there is can we start combining data sets together? And these could be from disparate institutions. And the other question one of our collaborators asked was, well, if we take samples that are otherwise pretty similar and we combine them, are the results that we get on one institution's DSP machine versus the other, are they similar?
I see.
So that's what we did. To do this, we systematically looked at different serial sections where we cut blocks of FFPE tissue. And we did this across 4 different tissues. These were colon, tonsil, and the germinal centers of tonsil, and in 2 different cell pellet arrays. And I could talk about that in a second. And the idea is we're going to cut these tissues and send them off to these different institutions and do similar profiling strategies. And in particular, we were interested in the profiling strategies of increasing the ROI size. So we vary that from all the way down to 25 micrometer circles, all the way to 500 micrometer diameter circles. We wanted to see, like, do we get similar genes above LOQ? We expect to see, like, the number of genes we detect increase as we increase the ROI size. We wanted to be able to quantify that. And in addition to looking at LOQ, which is like kind of like the background, like limit of quantification, we were interested in looking at raw data and the expression that we see from, say, one slide to the next slide. In addition to the varying ROI sizes, we were interested in not just like looking and comparing similar cuts, but whenever there's differences that arise from differences in biology, these could be comparing different cell lines, or taking two cell lines and having pure dilutions of each of those, and then also like maybe a 30, 70%, 95, 5%, where we mix those. And we would expect, but when we compare samples that are pure cell line versus another type of pure cell line, that we would see differences arising from the different biologies of the cell lines come out. Whereas when we like get closer and closer to like maybe 95 and 5%, then they would be very similar expression. So this is what we did. There's a lot of information right here, and for those interested, this poster is also available online as well. This is giving you an overview. This is a section, this is just an example image of the colon where we take 200 by 250 micron ROI sizes, and that's the ROI profiling for this. And this is just a breakdown showing that we have 7 different slides that we've cut, and these represent different experimental conditions and different institutions. We have technical replicates built in, and we also vary in 2 of the slides at 2 different institutions. We compare fresh cut versus taking the FFP sample and put it in -80 for 30 days, and then sequencing it. So we're trying to compare as many things as we can, while trying to keep the reagents and these types of variables as consistent as possible.
Now, quick question. We're using the Cancer Transcriptome Atlas. So can you tell me a little bit more about that? Like, how many targets is in that?
Yeah, absolutely. So with the whole transcriptome, you might be familiar with, it's over 18,000 for the human. We have 22,000 gene targets for the mouse, and those are unbiased. But for cancer, I think it's really nice in terms of throughput, increasing number of samples, to have a little bit more of a focused gene panel, and these include 1,800 targets. But these have some redundancy built in. There's multiple probes that we capture for each target, so it ends up being many thousand different targets. We tend to for simplicity for this talk, we'll collapse those down to those 1,800 targets. And we don't expect every gene to be expressed in every tissue. Exactly. So we will filter based on limit of quantification. So these 4 different types I have. Some tissues will have 1,000 genes above limit of quantification. We'll use those for the analysis, etc.
So what were the results? Can you tell us what we found?
Yeah. So actually, it was I think confirmatory. Honestly, when we look at overall the great concordance, I think tonsil is great here. So we took the germinal centers of tonsils. We profiled these at 25, 50, and 100 micron diameters, and it's if you look at the y-axis here, this is the per slide mean Pearson correlation. So higher is better correlation, and it varies based on target diameter. The y-axis here it's a bit like splitting hairs. So like the Pearson correlation is 0.975, so it's overall pretty high. We're able to do this both between slides. That's with this plot here, and as well as looking at within slides, how similar the results are when we look at within slides, which makes sense as you get smaller and smaller and smaller, just based on the spatial placement of the cells in there. We will get fewer and fewer targets in there, so we expect some decrease in there. It was actually surprising how high it was still, which is great. And this result is it varies from 25 to 100 microns. It's in tonsil. We also did a very similar thing in our cell pellet array, and there we varied 25, 50, all the way up to 500 microns. We're able to see that. And then in particular, the slide differences that we see between slide replicates are very similar. The 25, albeit is a little bit weaker, it's still pretty high correlation.
And I also see that the between institute correlation is actually quite high as well.
Yeah, so the between institute correlation is pretty high. It's whenever there's biological signal, there's not a lot of variation that's like specific to institution. And what I mean by that is, and here this is the 8-core cell pellet array, and these 8 cores, each core represents a different cell line. So these cell lines vary like different lymphomas, leukemias, carcinomas, melanomas. So there's different biology that's present there. And if we look at the top 10% of genes in terms of coefficient of variation, in this dendrogram here in this heat map, you'll see that these different colors here represent the different cell lines, whereas the colors in this bottom right here represent different slides. So we don't see a lot of slide-to-slide effects when there's dominated signal by the different biology. So that's, I think, represented here. And I like this how these different, it's really discrete. Like here's these different sections that are highly expressed in one cell and in the next. And I've also like labeled some genes of interest that might be specific to the cell lines.
So this seems to suggest that if you want to do spatial transcriptomics and you're working with a lot of collaborators, multiple institutes all over the world, that you can be confident that the GeoMx digital spatial profiler gives you really reliable and reproducible results. And I think this is particularly important, say, if you're analyzing samples from like a clinical trial.
Yeah, absolutely. I think, I was actually kind of surprised by this. We picked 25 microns as sort of like, all right, that's really too small. That's going to really be discordant. And I was actually like underwhelmed how discordant it was. So yeah, it's great.
Excellent. Well, thank you so much, Tyler.
Yeah,
and
thanks.
if you're interested in learning more about the GeoMx Digital Spatial Profiler, Cancer Transcriptome Atlas, Whole Transcriptome Atlas, please come see us during this week at AACR at booth 1719. And as Tyler mentioned, you can find a digital download of this poster as well. And we hope to see you at our poster sessions. Thank you so much, Tyler.
Yeah, my pleasure.